At least it's something like that, you know. Than the 40% after they got there. Yeah, I don't know. That's what they said. She's very in a lot of Makes sense. I don't know. Like you're saying that, but I'm like there. Like that. Whether or not something else you want to pay is not sure. We don't believe it started. Oh, yeah, it's too long, as we talked about. Oh, so kind of, we don't have from there. So, yeah, okay.
Very good. Okay. Okay, so good afternoon, everybody. Can you hear me? Yes? Is it not, Carolina? Is it good? Very good. So as people are getting their food, I am going to start with introductions, but it would be good if you are a little bit quiet when you get the food. Otherwise, wonderful. It's an incredible honor to introduce Daniele Fallin, the current dean of the Emory School of Public Health at Emory University. So it's amazing to be able to let you know a little bit about Dr. Fallin's trajectory, incredible trajectory in this field, in the field of public health. She got her undergraduate training, her Bachelor of Science at the University of Florida, Gainesville. Then she did her PhD in genetic epidemiology at Case Western Reserve University. And then the Johns Hopkins University did an incredible job with attracting this amazing talent in the Department of Epidemiology, in the genetic division of the department. And I have to say, I was a student of Dani for only a few days because I wanted to study genetics, epi. I wanted, honestly, I didn't care about genetics, but I knew it's important for the environment. So I said, I've heard this amazing class. She was teaching this amazing class in genetic epi, so I wanted to take advantage of that training. Unfortunately, my father got sick that time, and I couldn't complete the class, which was one of my missed things during my training as a PhD student. But I had the privilege to work with her later on. And none of the epigenetic work that we've been able to do in the Strong study would have ever been possible without Dani. So thank you so much. You had an incredible impact in a lot of the work that we are doing now. And I think that says a lot about who Dani is. She's such an incredible, generous scientist. She's always looking for real, high quality, impactful work. And she doesn't, she gave us so much. And she's like, it's okay, you can continue doing all of that work, because she's became very, very busy. She became the chair of the Department of Mental Health at Johns Hopkins University. And then I was thinking, oh my goodness, that department, they're so lucky to have Dani as a chair. And then she became the dean of Rollins School of Public Health at Emory. And I was just like, oh my goodness, these people are so lucky to have Dani. And it's been amazing to have you as a role model in many ways, balancing science and personal life. And so thank you so much for joining us today.
Here, thank you so much, Anna. Thinking about people who are lucky, you guys are so lucky to have Anna. We all need this kind of cheerleader in our lives. Such a thoughtful scientist while also being such a wonderful friend and mentor. And we did have a lot of fun in the early days of figuring out what could be done in Strong Heart together. And it is unbelievable to see how that work has progressed and all the folks that you've recruited that are just amazing. It's my privilege, really. So thank you all. We were talking last night about how I would like to move to having scientific talks only be 30 minutes. And yet I have a 50-something slide presentation and short in time. So I may just skip some if I see folks in the room that are like, yeah, then I just keep going. And so those of you online, and I did see some familiar friends online. So hi, everybody. That's why. There we go. Just up front, I think, Anna said, I've been dean for 8 months. I am still figuring out my footing of how much research versus other stuff I can get done. But I have the privilege of having a really remarkable team at the Wendy Klag Center in Baltimore, led now by Heather Volk and Chris Ladd-Acosta, who are at the top of this, but also with Aisha Dickerson and Kelly Banky spending a lot of time together. And so the 5 of us really work together super well, and they are just incredible researchers themselves. And so a lot of the work that I'm going to show you, which while may have been started by me, I think is led more day to day by some of these folks. So I want to acknowledge that up front. And, I started, the title of this talk is really my own personal motivation, which is my work has been in autism and developmental disabilities. And that's the lens through which I got interested in epigenetics. So I will use this as the frame, but many of the examples that I will give may not be autism specific. So I won't say much about autism other than this is a developmental disability that's characterized by social impairments as well as kind of neurological issues like repetitive behaviors. And its prevalence in terms of diagnosis continues to go up in this country and around the world. New estimates from CDC came out last week. So we are now at amongst 8-year-olds, 1 in 36. So relevant public health problem, not today or after this, we can talk about what that prevalence increase means and doesn't mean. But suffice it to say that I think, the impact to our society is large. So, as I said, I got trained as a genetic epidemiologist, and so from the beginning, I was very interested in kind of pure genetic causes of things, but recognized early on that that is a myth. That in fact, all genetic relations to outcomes have some interplay with environment. And in autism, there really had been this legacy of folks who were doing genetics and genomics work very separate from the folks doing environmental epi work. And that's, again, another conversation for a longer time about why that happened and why it was perpetuated. Now, I think there is starting to be a recognition that these two fields should come back together. This is a fairly old graphic, but I think still useful in talking through both of those fields and how those fields could come together. And while that is changing, I will tell you, the landscape for actual G by E papers in autism is still, it's on a slide. So we're still not there, but just to give you a glimpse of, you know, for those of you who know the genetics world well, all kinds of different genetic variants, rare, common, single variant, copy number, all those things have been looked at. And in environment, at least in terms of the G by E, things that might be familiar to many of you in terms of air pollution, vitamin intake, and folic acid pathway, and then other things like experiences during pregnancy, like infection, even other metabolic disorders during pregnancy, that kind of thing, just to give you a flavor of those. But I put this in here just to give you the motivation of the rest of the talk, which is when I got interested over decade and a half ago in this, what became apparent when I learned about epigenetics was that this could be we'll talk about either a mechanism or a biomarker that's relevant to this whole picture, that's relevant to the genetic risk side of things, that's relevant to the environmental risk side of things, and that's relevant to their interplay, and may be a helpful tool as we go forward. So what I'm going to then share with you is how I think it's a helpful tool. So what the utility of measuring epigenetic marks, and particularly I'll focus on methylation, would be for epidemiology broadly, and then in my motivation for autism epidemiology. So with that, here's kind of the roadmap of what I will push through pretty quickly. And again, I'm sorry if the pace goes too fast, but I want to make sure we get through. So a little bit of an introductory to epigenetics, and then a little bit more about this utility concept. And then I'm going to walk through some examples that build off of that utility concept. So that's what will happen when I start talking about the different ways in which things can be associated with methylation, then what that means for us in epidemiology in terms of both mechanism and biomarker utility. And so normally when I give a talk like this, I'll spend about the slides talking about epigenetics. I'm not going to do that here. All I'm going to do is show one slide to prompt me to say, for those of you who do understand epigenetic and epigenetic marks, that covers a broad landscape of things that are really important. I'm only going to be focusing on data that come from DNA methylation. That is partly because of the tools that are available, and partly because for stored samples in large epidemiologic studies, methylation is fairly stable, and so it has been a useful tool. But ATAC-seq and other things also relevant, and certainly if you have fresh samples, all kinds of other chromatin marks and things like that can be important. So I don't mean to be exclusive to methylation in what I show you. And I put this one in here too. I think probably everyone in this room recognizes it, but it's always important for me to remind myself, you know, I do believe and care about inherited sequence level variation, but generally speaking, that's true from birth and throughout your life course. Whereas we know that basically every human condition has a life course component to it in terms of how it ebbs and flows and what the consequences are. And epigenetic regulation does have this kind of ebb and flow itself. And so I think it's another useful frame is to remember that this is a dynamic system. So the first time I heard this concept was from a guy named Andy Feinberg, who was at Hopkins, and he said, "Oh, I'm looking for somebody that can work on epigenetics." And I thought he meant epistasis. And I was like, "Oh, I can do 2 genes at once or more, you know," and he's like, "Yeah, that's not what I mean." And so then I educated myself and learned. And so one of the first things we did was write this little framing paper together with Hans Bjornsson, who is just a fabulous researcher in his own right now, but at the time was a doctoral student of Andy's. And I kept just asking from an epidemiologist question, like, what things do we know control epigenetics? How might epigenetics relate to disease? Where does it fit with the gene and the environment and that kind of thing? So we wrote this piece a long time ago now. And It's evolved since then, but it actually wasn't too bad. Now, when I make this articulation about utility, here's kind of the way that I pitch it. And I think of both mechanistic insights that we can gain through this kind of work, but also biomarker utility. Even if an epigenetic mark is not part of the pathway to a particular disease, it may be useful in some settings. And so I'll elaborate a lot more on that. What I do want to use this slide for very quickly is to say there are probably even more than these, but this is the way that I and our group has been thinking about it. And the first is, if it's mechanistically involved, it may be a mediator. So a genotype or an exposure, remember both sides may influence the epigenome in ways that then influences disease, right? Or mediation. It may also be an effect modifier. And so think of this like imprinting. So the idea of you have a genetic thing that can cause a disease, but it only ever happens if it's turned on or off, right? And so that's an effect modification kind of scenario. And then people in the lay literature talk about, oh, epigenetics, that's the study of gene and environment, right? But to me, not necessarily that, these are kind of gene flow or environment flow in terms of risk. There are situations of true gene by environment interaction where, where genetics are effect modifiers of environment risk or the reverse, right? But that's a little bit more complicated scenario. So we can do that as well. But I think we have to be honest about what we're actually trying to get at. And then a little bit later, I'll talk about even when there's not these kind of mechanistic scenarios where these things could be helpful. And I'm not going to be able to talk through examples of all of this today, so I'm just going to shrink it to this part of that. So I'm just going to talk a little bit about the mediation concept and break it apart. Mediation of genetic risk, mediation of environmental risk. And so to the roadmap, the first thing that I'm going to need to do is convince you that there are reasonable associations between this part of that mediation scenario and certainly the biomarker one too. And for me with ASD. But then remind you of this association of genetic relationships to epigenetics. And then lastly, of this association, which I think is probably familiar to everyone here. So that's what these first three things in the roadmap are. And then we'll go back and talk about some examples of really the full pathway and then some examples of biomarkers. Make sense? All right. So as I said, autism was the motivation for me. So Chris Ladd-Acosta, who was a student of Andy's at the time and then became a postdoc with me and is now an associate professor in Epi at Hopkins, joined with us to look at epigenetic marks as associated with autism in the most obvious tissue that we thought of at the time, which is brain tissue, right? And so I'm not going to spend a lot of time belaboring this point today, but we could certainly talk about it at some point, the tissue specificity issue and the challenges around this for the kinds of work that many of us who do epigenetic epidemiology do. So we started with brain. There are so many challenges, right? The first is the autism brain banks are very, very limited. They're at all ages of death. There's all kinds of issues that came into cause of death and that kind of stuff. And importantly, what part of the brain should you be looking at? So there's just, layers of challenges. That may be true in all the things that we do, but it was particularly problematic here, and particularly problematic to see things at the end of life when we're looking at these kinds of epigenetic marks versus at the beginning of life. And I sort of would want to lean towards that. But we did it, and we did see some associations, and some of these, I will say, have come up now in things that we've done in more modern times. So that brain work was useful and helpful, but was limited. Separately from that brain bank work, we have a cohort that's called the early study and early autism risk longitudinal investigation. And this is the enriched likelihood cohort. So these are all families who have a child with autism as they go through a subsequent pregnancy. That means that the expectation for autism cases is more like 20% than the 1 to 2% that it would be in a general population. So that makes doing a prospective cohort and expecting enough outcomes possible. And I just gave you a little bit of sense. I'll show this again later of all the things we did. So we recruited in pregnancy. We did lots of biosampling and other kinds of measurements during pregnancy, at birth, and then through at least age 3, where we could do early life developmental assessments. They are now being followed as part of ECHO. So they're now into school age. I was talking with someone earlier, they were getting their teeth to go to Manish and all that stuff, right? A similar study, almost exactly analogous in terms of design, was happening at UC Davis, the MARBLE study. So this was Irva Hertz-Kyoto as PI at the time, Rebecca Schmidt is now. And in fact, Irva was also an early PI because early was at Philly, Davis, and Kaiser, Northern California. So we have this great design, 2 studies that can come together. And for the purposes of what I'm showing you, we had samples taken during pregnancy, dad's semen and mom blood at multiple times. And then at birth, placenta and cord blood, and then certainly things through childhood as well. But those are the relevant ones. And then I should also say, while I focused here on what kind of methylation measurements were used, we certainly also have measured exposures like metals and other things at these in these same samples at these same times, and also have other matrices like meconium and urine and other stuff. So in terms of ASD and epigenetics, there's multiple choices you can make. So one we really cared about was looking at placenta. And so this is work, I'll show you in the next slide, with Janine LaSalle's group and folks at UC Davis who are part of the early Marvel's group, and funds that we have together. Just looking at what we can see in terms of placental methylation and ASD outcomes. And indeed, we do see that there are associations in this particular paper in the 22q region that are replicated in other data sets and do seem to be specific to ASD versus other kinds of developmental disabilities. So that's useful and important in one avenue of further discovery. At the same time, I mentioned we had semen collection. So we also looked at methylation in dads and found that at least for a 12-month presentation, we could see associations between methylation spots. And this is the sort of some of the top hits, and then this is blowing one up so that we can see it well, where we could see kind of quartile dose response looking measurements of methylation for a region at a time, or what we call differentially methylated region, in dad's sperm associated with what happened to this baby 12 months of age. We have just done analyses for this at 36 months, and stay tuned, that's coming out very soon. That also shows, I think, a continuation of this thinking. And I think that's important because we often, even me, we think about maternal child dyads, and we really think about mom's exposure and mom's stuff. But this, I think, would hint that we need to be thinking about dad's exposures too. So then really, we have all these multiple tissues, trying to put them all together. So Kelly Bukowski, who was a postdoc of mine, who's now faculty at University of Michigan, took all these together. So took the array methylation data that we had from placenta, from cord, from mom's blood, and then looked at 36-month outcomes. And what she saw, I think, was really important and interesting. If you just look at the kind of nominal hits, so if you just look at not something that made genome-wide significance, but just say like at a 0.05 p-value in terms of any CPG, if you look at that and you look in known autism genes, so there's a Safari database that's a curated, updated database of evidence for autism genetic risk. If you look in those genes, she saw that there was, for each of the different tissues, there was enhancement for autism genes where there would be CPGs associated with 36-month outcomes, right? And then what she said is, and how much do they overlap? And so this cute little star is then showing, if you take those where there's some evidence in each tissue of enrichment, what does that overlap look like? And it's not bad. And what we took from this is, again, whether or not we're talking ultimately about mechanism, we're getting signatures that are detectable across these different time points and across these different tissues. So I hope I've given you a flavor that yes, what we don't know is whether methylation is leading to autism or autism is leading to these methylation changes, although I showed you some different time points where maybe we can lean to the top versus the bottom scenario. We at least know that there's robust associations across tissues and matrices and kinase. So then the next question was, what about association with genetics? And I'm not going to believe at this point, because does everybody feel comfortable that genes are associated with methylation? Okay, great. So early on, we were doing this with Andy Feinberg's group with rheumatoid arthritis. And this was the early days when you're still trying to map these, what we call MEQTLs or methylation quantitative traits. They look something like this, where you can see that by genotype, you have these different distributions. And importantly, if you look spatially, it looks something like this, that SNPs are the kind of lower level dots, and then methylated positions or epigenetics are the top level dots. And you can see that there's a whole bunch of correlated SNPs that will associate with methylation at a particular CPG or at a smaller correlated cluster of CPGs. So these correlated methylation units, and you have these correlated SNP units, and they tend to associate, right? And that's useful for many reasons, but I'll show you the ways in which we have used it. So that's the genetic association. There's a robust literature now on building MEQTLs per tissue, MEQTL maps per tissue. And then on the epigenome on environment side, I think this audience is probably even more savvy than me. I just put this in here to remind me to say, we see it across, all kinds of exposures, lifestyle ones up to toxicant kinds and everything in between. And this was Kelly's brilliance again, when we wrote this review a long time ago. She was like, I hear you, Dani, you talk like an epidemiologist. The toxicology people don't believe any of this. So like, how are you going to do this from the tox side? So when we built this review, we tried to build a literature that was toxicology literature and align it with the literature that was epi literature. And this is old. We should do this again to have a much more updated version of this. But I thought that was important insight in her about bringing these two together. So quick landscape of those associations. So the components of these things. So now let's talk about mechanisms. So the idea would be for epigenetic mediation of genetic risk, that these genotypes that I showed you that can control methylation, that it's that methylation change that is somehow then part of the pathway to outcome. And so one of the first times that we tried this was with rheumatoid arthritis. And so this is that same figure I showed you before, but we followed something called the causal inference test, and we can all talk about whether that's the modern thing that you should be doing. But at the time, this was one way of doing mediation analysis. And it had several components. So the first was to make sure that red arrow existed. And the next was then to make sure that the epigenotype to outcome existed. So this is that association. And then to make sure that the genotype to outcome existed. So that's that part. And then, and we could see it easily, and this is for a particular CPG and a particular SNP. And then to look at what happens when you look at that genotype to disease and you then remove or include the epigenetic part. So this is, I think, classical concept of mediation, no matter which tool you use. But that's the rules and the sort of stipulations at each point were specific to this genetic application. And so we could see exactly this, that there seemed to be evidence, at least at this level of mediation. And I'll remember the time we were trying to will this work when we move to an environment? Is it the same model? Do we need a different model? How's this going to go? Yeah. And then I bring this up because I think there's good evidence for mediation in rheumatoid arthritis and now in many genetic risk stories. But it was interesting at the time, and I think still not as totally appreciated, that we could be talking about different gene sets than we think. So the SNP and what we would learn from genetic discovery may be somewhere like, say, where that red dot is, right? But the methylation that it's controlling, that is part of this mediated pathway, is way over to the left, if you see that on here, which is part of regulation potentially of a different gene. And so when we're thinking about the functional biology, it may be more useful to think about that gene that's being regulated rather than the gene where the SNP is or is close to. And that was, I think, a fundamental challenge that wasn't really the way that we were thinking about things prior to evidence of this kind of mediation. And so in autism, we've taken that same approach. So here's an example of where there was a GWAS ASD hit in a gene called PPRA3. And then we had these SNP map, MEQTL maps. This one I think is the blood map. And you can see where the strongest associations are. If you then drop down where those CPGs are versus where that SNP is, very different genes are potentially in need of interrogation, right? Does that make sense to everybody? Yeah. And so we were able to do that not only with blood maps, but we had the opportunity to do it with cord, with field brain, with placenta because of that data set that I showed you. And so we can see some of this is because of sample size, that some of the maps are not as good as other maps, but you can see certainly with placenta, just knowing that hit on that gene didn't tell us a lot because it's with methylation and potentially other regulatory marks in the whole region. So hopefully that gives you a sense of just a little bit of the utility of why this could be useful when it's a potential mediator of genetic risk. What about for environmental risk? And I think this is a clear example, so I just put this in here. It's not our work, and we made a comment about the work, but it was something very interesting to me, and I'm going to talk about smoking a little bit more. But there was a AJE paper that looked at maternal smoking and whether or not placental methylation was a mediator for birth weight, and I think they did a good job. I think it's a good one to look at if this is of interest to you. There are certainly some caveats, but I think it was good. I'm going to give a shout out to Artha, who's done a lot of this kind of mediation work with much better mediation tools than our original CIT, but also with longitudinal time to event data where hazard estimation was possible. And so in this case, looking at whether arsenic exposure and CVD outcomes of both events and death were mediated by particular methylation positions, and did a beautiful job of showing indeed that's true, and you can replicate it in other cohorts. So really lovely stuff. And I get the privilege of still giving advice sometimes. And they had this really cool graphic that when you first look at it, I was like, what does that mean? And then I saw it has every component of the findings. It's really well thought out. So I'm going to get back to environmental risk when I talk about biomarkers, but I want you to think about sometimes when I'm talking about biomarkers, it doesn't have to be mechanistically involved, and it also means that it doesn't have to be the tissue of specificity for mechanism, but often these are hinting back towards mechanism. So it's hard to make this dichotomy of showing you what's an example of environmental mechanism versus what's an example of biomarker, because it's still kind of fuzzy days, but that's why I set it up this way. So now we're going to leave the yellow and go to the blue. And I would argue to you that this is actually where it's been most surprising to me of how useful this could be. And thinking through, what if the epigenetics is just a consequence of exposure, but has nothing to do with how it causes disease? And then we've kind of known for a while that sometimes epigenetic marks are consequences of disease state, but that may still be useful for diagnosis, prognosis, other kinds of things. So I wouldn't be too purist about that. But I'm going to jump into the story that hopefully most folks know about, I think, just so telling and convinced me that this biomarker of exposure world could be an important one. So how many of you know of the prenatal exposure epigenetic signature concept? Yeah, Andre and Anastasia, yeah. A couple others do, yeah. Okay, so I won't spend a ton of time, but since several of you may not have known this story, I will slow down a little bit. So in early days, Stephanie London's group and Bonnie Javert specifically, who are at NIEHS, were asking the questions in the MOVA sample about whether or not, if you looked at methylation in cord blood, could you see associations with which moms smoked and which moms didn't smoke during pregnancy? And they had cotinine measures, I think at trimester 2, as their measure of smoking. And so they did what early days of what was called an EWAS, or an epigenome-wide association scan. So that's what you see going from left to right is all the chromosomes looking at CPGs. And they saw 26 sites that met genome-wide significance for association between cotinine levels, so smoking, yes, no, in this case, in the first analysis, and methylation at these particular sites, so at these 26 particular sites. At the time, no one else had these kind of data, and they wanted to replicate. And so that wasn't possible, except that they collaborated with the folks who have the NEST cohort in North Carolina, much smaller cohort, because this was 1,000, a little over 1,000, I think at the time. I think the NEST one was under 100. So you'll never get p-values to be able to replicate, right? So what they did instead, which I think was really valuable, is they did the same analyses. And here's just a representation of what it will look like. Here's a single CPG. Here's a distribution of exposed versus unexposed that you might be able to pick up, right? They said, let's plot the effect size. So this one, is looking like it's the unexposed are lower than the exposed. So that's a little bit lower than 0 to show you that effect size, the difference between those two means, right? So they said, let's plot the effect size in our discovery sample and in the NEST cohort. We know we won't get p-values that are statistically significant because of the sample size. Look at those effect size comparisons. So this is for each of those 26 CPGs compared to, and I don't think NEST had cotinine levels. I think it was self-reported smoking. I might be wrong. Somebody knows that data better than me. Remarkable, right? Like, when do we ever see that kind of replication? So then Chris Ladd-Acosta, who was working with me, we had the same kind of data on 3 to 5-year-olds. And so we asked the same question later. Remember, this was cord blood. Can you still see it into childhood? And pretty close, not exactly the same, but pretty close. So around that same time, Allspac started doing this, and several other folks now started having these kind of data. In the meantime, PACE now has a much larger sample set. So things are building, but at this time, it was really like remarkable to think these signatures, if you took those 26 and you built a kind of predictive model of whether or not that child was exposed or not exposed in utero or smoking, you could do really, really well. And I'm not going to show you those ROC curves, but they're really good. And that to me was remarkable that you could do that in a 3 to 5 year old without actually having pregnancy data. And then Allspac showed it at later ages, and now other studies have shown it. And so even into adulthood, you can see these prenatal exposure signatures with pretty good accuracy, and they are separate from self-smoking signatures. I didn't give that work, but there's separately one that you can build for self-smoking. And so much so that now there are these kind of polyepigenetic scores that you can use to predict. Predict is a weird term too, because you're thinking backwards, but statistically predict a previous exposure. So I got really excited. I think Chris got really excited. What could this mean for all these epidemiologic cohorts that didn't measure exposures of interest to us, and have banked samples, but nothing else, right? Can we get these kind of exposure data for UK Biobank, for other things that don't have these things, right? That's when we had a lot of conversation with, well, you still have to build the reference. So, we had the beauty of starting with measured smoking that people believed in, to make those first reference panels. We don't always have the privilege of that, but that's why I got really excited. And so then Chris has taken this and said, okay, well, proof of principle, if we were to do an epidemiologic study where we didn't have the exposure of interest measured and all we had was the methylation, would it be as good? So she worked with the ERIC cohort. How many people in here know what the ERIC cohort is? Most people. Okay, great. So she worked with the ERIC cohort who have cancer outcomes, and had done methylation array measurement on all their sample. And this is the kind of associations you get in terms of hazard ratios for their black and white samples, because ERIC has sort of two different pieces to it, for several cancers. You can see them there. And this is self-reported. So this is like what they had normally, right? This is the same picture if you just use a methylation predictor of from their blood taken during, adulthood, right? Well, in this case, I should say, in this case, this is self-smoking, so that makes more sense. And then, so the first thing to say is, cool, that does mean we could probably use this as a pretty good biomarker if there ever were something that isn't measured, and still be able to do the epidemiology. Even if it's not part of the pathway, it's still giving us more information on risk. And then she did, I think, what was really important. What if you controlled or adjusted for self-reported smoking? And she sees that you actually still get additional estimate of hazard ratio even after that, right? So methylation may actually be capturing misclassification from self-report, other correlated exposures. I don't know, you guys might have better ideas than me. But I think this is really telling for the field of epidemiology when you start thinking about using these signatures of exposure, if we can build them for other exposures beyond smoking as we move forward. So I think it's a really cool proof of principle. So we are doing this for other exposures like many of you. So this is just showing that you can detect associations between methylation and air pollution. This is global methylation, but you can also see it at single sites. Here's an example of at a single site. And we see this in both placenta and cord blood. So many of them are, again, cross tissue, giving you similar answers. So as a biomarker, I think that's a relief, frankly. And then we've also done it with prenatal vitamin use. And I put this slide in here just to say, this was interesting. So this is John Dow's work with Kelly Vokulski and others at University of Michigan and our team. And so this is in that early data set looking at prenatal vitamin use and methylation in cord blood. And I won't belabor these details. Basically, it's looking at 3 ways: multivitamin, folic acid supplementation, and prenatal vitamin as different kinds of exposures along the same lines. And just look at the left first. These are plots. I don't know how many of you are used to seeing these volcano plots, but sort of what's going from bottom to top is the p-value, if you will, to more statistically significant versus left. And then left to right is the effect size. So going to the left is hypomethylation, going to the right is hypermethylation. So you can start to see in these cases kind of a general shift of hypo versus hypermethylation across the genome for these. And one of the questions we had, because the question was about, it really started with folic acid supplementation and knowing that there's this, methylation pathway issue that we need to pay attention to for this particular exposure. So we looked at NTHFR genotypes at the same time. And so this is what we would get overall. But really, if we separated by carrier versus wild type, we saw very different patterns in terms of hypo and hypermethylation across the globe. And then this is kind of small sample sizes, but you can see for the wild type in both groups, we see that association. We didn't see it for the carriers, right? So I put that in there to say we're building these predictors, but to Anna's point from a conversation earlier this morning, there's so many complications to this that one kind of heuristic is not always going to work, and we have to really take into account things in this case like a background genotype. And I put this one in. This is a follow-up, slightly different exposure windows. One was 3 months prior to conception, and this one is in the first month of pregnancy. And it's just one of those 3 exposures. You can still see differences here. We had placenta as well as cord blood. That previous one was just cord blood. But what I want to say is, again, we're thinking kind of maybe of building biomarkers, but we're also thinking about, does it give new mechanistic hints? So don't think that I'm arguing only one or the other. And so in this case, John took those hits and looked at suggested biology, and lo and behold, behavior, neurodevelopment, these kinds of things in this group came up. So maybe we should be doing more mechanistic mediation questions next in our data set based on this. Okay, so that was of the kind of exposures we've mostly been looking at. Recently, I had a fellow who was interested in social exposures and methylation. So we got a little bit into this ballgame. It's a little bit new to me. So this is Megna's work, and she started by just saying, look, Dani, there's already this building set. We could be building potentially methylation signatures of social determinants in ways that we don't have access to data for some of these populations. And so she did a nice job of characterizing the complications that arise, like the timing of the exposure of some particular socioeconomic position indicator, right? And also the timing of this biosample that you took. It's very complicated to think about how you match all those and show where the evidence is and isn't in that regard here and here. And now we're using those to see whether or not they would be much like that lung or the broad cancer proof of principle, whether or not these predict neurodevelopmental outcomes as well as the actual data itself, so that we can, the SEP data itself, so we can see could this be useful more broadly. And I should say, Aaron Dunn's group at Harvard is doing very similar things if this is something that's of interest to you. And I think there's good reason to believe this is going to keep happening. That review is only like a year old, but I think these things keep coming up. So the biggest challenge that we have in their cross tissues, the biggest challenge that we have in that one is measures of socioeconomic position are all over the place. So you need your deep bench colleagues in thinking through that kind of measurement and what it's capturing and not capturing to think through whether this is going to be useful. So I would say jury's still out, but I want to share. So in summary, I got excited about the measurements of prenatal exposure and association with methylation at first for mechanism. Now, I think really for all these other biomarker utilities that we might be able to get after, although I'm not giving up on mechanism, but that's where we are. And I think it's really cool that you can capture prenatal things later in life, but it's also cool that you can capture in life things. So both things are true. Okay, so then I didn't put much slides in here for health states. In fact, I don't think I put any in there, but I don't want to lose track of, absolutely, there's a field of biomarker work in methylation that is about disease states. So some of those ASD associations that I showed you before, if they turn out to be that it's the fact of experiencing autism that is creating these methylation changes in placenta or in later cord blood or in brain, right? That is still wildly useful in our field because we don't have good biomarkers of diagnosis, right? Cancer has done some of this for sure. Other areas have already done this. So don't lose sight of that. It's also very helpful if you're thinking about accommodating co-occurring conditions, and you may not have the clinical data on, diabetes and obesity and a particular type of cancer and this and that in a cohort, you might be able to use some of these signatures to estimate that in the context of what you want to control for or how you want to look at multiple outcomes, those kinds of things. So I think that the opportunity is just as good on the disease biomarker side as it is on the portion called biomarker side, exposure biomarker side. Okay. So then lastly, age. So I follow Twitter, and John Greeley just posted yesterday. Somebody remind me again why we do these epigenetic clocks. And I was like, oh crap, that's tomorrow. So hopefully many of you have heard of this. This is kind of the seminal set of papers that showed in adults that much like we can find signatures of smoking, you can find signatures of chronological age. It's the same concept. You just, from the whole set of CPGs, you're filtering out the ones that seem to be most associated with some outcome, in this case, age, right? And then you're building a predictor based on that. And one of the things to tell you, it doesn't have to be the same predictor. Like different ones have different subsets of those CPGs, and it still performs pretty darn well, right? There are lots of nuances, but that's the concept. And then where folks have gone with this, apparently in the forensics world, this has been wildly useful for predicting age of folks that, pass away or something like that. Just the straight prediction of age. But where it's been most useful, or at least presumes to be most useful, is in age acceleration or deceleration. So this residual concept of here's what we can predict and it looks pretty good, but when you are off of that prediction, high or low, does that imply accelerated biological aging or decelerated biological aging? And that's then the measure that's then used for asking questions about health outcomes. So I got engaged through Andy and others with the group doing some of this work on mortality early on. And I think it is true that the Horvath clock and others did a good job of predicting mortality. So that's a gut test that, yeah, it's measuring something by having this accelerated aging versus decelerated aging versus just age itself, right? There's something to that. It's along the telomere story at one point. You could see it in multiple cohorts. So I think that's true. It is now started to be used for things like those social exposures, and that you can see social exposures having an impact on this measure of acceleration or deceleration. That may be useful and meaningful. We need to talk through exactly how and when. And then most of these have been adult based, but there are now pediatric clocks and there are gestational age clocks. And so given our interest in autism and sort of what happens during gestation and then early life, we have used both. Ashley Song, who is a postdoc and I think about to be faculty in our group at Hopkins, started tinkering with this in our early and marbles populations. And so, she built, there's two kind of competing gestational age clocks. She built both to see how they performed in our data, and then could build aging acceleration and deceleration off of that. And one of the first things that she's done is just say, okay, what about exposures of interest to us? Do they seem to affect aging? We've already showed you that they affect just methylation amounts, right? That was some of the stuff I showed you earlier, but do they affect this acceleration or deceleration in terms of gestational age? So here she's showing, if you look at a air pollution lag model, you can see where there does seem to be aging deceleration. This is gestational age, right? As associated with exposure windows going from right before pregnancy through pregnancy. So in addition to just amount of methylation, it does look like deviance from expectation of methylation may be associated with air pollution. And I think others are trying to do these same kinds of things. I'll give John, credit. We don't know what this means and how to exactly use this moving forward. But it's pretty consistent that it comes up. And for us, deceleration, not developing as quickly as one should, is consistent with what's going on in developmental disabilities. So that might be useful. Okay, so that was the main part of the talk. And I want to leave time for questions. What I had put in the rest was just some real caveats, like Kelly Kulsey's team is doing a lot of work of single cell and cell sorted placenta work, because the placenta data that I showed you is bulk tissue analysis. So she is now going back, her group is now going back with us, and particularly a student named Kyle Campbell, looking at if we are able to use the reference panels that she has developed through careful work, can we deconvolute and adjust for cell type proportions in our aggregate placental data, much like we've done for cord blood and for other things. And so I'm really excited that those references are now available. We do see the ability to then see what cell type proportions are associated with exposures and outcomes, and that alone is useful information. And then we're able to adjust for it. And fortunately for the placenta work so far, we do see that when you adjust for the cell types, you actually see similar signals. So it doesn't seem to be driving some of that, but we also see that there are particular cell types that are implicated for particular outcomes. So I won't go through all those data points so that we have just a couple of minutes, but that was the idea, is to say it's not all rosy. There are many of these things that we need to think about at a deeper level, but I wanted to give you the kind of big picture. This is where I think the utility is. And I only showed you examples for these top two and this bottom top one. So there's much more that we need to think about when we're really digging into G by E and when we're using disease biomarkers. So I'll stop there.
Questions? You can slow on it.
That was phenomenal, really. I learned a lot and I enjoyed a lot what you said. I think the question is that you focus a lot on biomarkers, and you mentioned one practical utility on forensics. But I'm thinking in terms of public health, I'm thinking what would, if we think about 10 years or 20 years from now, thinking that all the caveats are solved and the biomarkers can be used on a wide scale, what would success look like? I mean, how do you think these biomarkers can change the way we practice public health and how we use them?
So again, think about something like UK Biobank or any of these other, or some of our own ones, TopMed, other ones that have this enormous amount of genetics data and are asking all these great questions. Their environmental data is small or non-existent in some cases, right? We had really good signatures of specific metals exposures. If we had really good signatures of other kinds of toxic exposures, or of potentially social determinant kinds of exposures, you know, you name it. If like you said, if we could solve that problem of building those signatures and then had them, we could open up all kinds of new questions and answers that would inform what we think we should be doing next for preventive interventions. I mean, it could be, I think, radically changing how we do epidemiology, particularly for those cohorts that were built as genetic cohorts and didn't have this other information, because they do have the biosamples, right? So that, you know, they can measure and have measured methylation. So that's my excitement, if it's possible. Yeah.
Thank you, Dani. This is amazing, and it's thank you. But I wanted to ask you about the causality and the mediation, you know, which is so difficult to do, and that at the end of the day, we don't really know: is it truly mediation? Is it something? Is it reverse causality? Is it you know, confounding? Is it something else? It's so difficult. So, what do you think of the Mendelian randomization tools? Are these helpful? In which way are they helpful?
Yeah, so does everybody know what Anna's asking in terms of Mendelian randomization part, right? So that causal inference test was based on that concept, right? It was saying that the genotype to epigenotype and genotype to disease is essentially unconfounded except for ancestry, because it's a genetic question, right? So it came from that world, right? And so I think we have been trying that. To me, that's one tool, and it's not a tool we can use when it's environment mediation rather than genetic mediation, right?
That's my question. Can we apply it really? I mean, people are applying it, but is it really working?
Right. So I think for me, it's more, we have to go back to fundamentals of how we thought about mediation for anything else, right? Which is, what does a temporal relationship look like? Is it biologically plausible? You know, do we really think if you perturb that mediator, would you change the outcome? If that's not true, it's not useful for that question. It might be useful for other questions, right? And so I think some of that we can do epidemiologically, and particularly in beautiful cohorts that have longitudinal data where you can have this, you know, question rather than cross-sectional analyses, which is what some of these are, that RA data was, right? Also having, bench colleagues and other folks who are thinking through these particular mechanistic challenges and can show it experimentally in ways we just can't in epidemiology, even beyond observational epidemiology,
So
There's gonna
something
be
that I am really worried myself is that a lot of these DNA's methylation signatures are really reflecting these early disease processes, which is what you show, with disease influencing DNA methylation. But I think it's exposures affecting disease, affecting this early DNA methylation that is related to disease. And that I think is what we might be capturing. That's my kind of thinking. And even with the environmental exposures, when we measure them as biomarkers, I am very worried that even in perspective, in beautiful prospective cohorts, all what we are just capturing is just early influences of the disease in the metabolic pathways and in the DNA methylation and other processes. So
it's possible, which is that biomarker figure rather than the mechanism figure.
Which is
Absolutely.
great if you interpret it as a biomarker diagnosis process, but that's not what we are doing in our field.
Yeah.
So I have the feeling that when I retire, and I know that I've been very philosophical about that, but I am going to be thinking that all the work we did was kind of useless. I am afraid, not really.
Not true.
We are learning a lot along the way, but the more and more work we do, I am the more and more worried that there is a lot of early disease processes that explain a lot of the work we do, and that we are not conceptualizing it enough, maybe because we are worried about the implications that it has.
Yeah, I mean, where I would come back to is the reason for me to understand mediation is to have a more proximal intervention opportunity, right? So in working with bench colleagues and others, is it possible that if you perturb that, the outcome changes? If not,
Recording in progress.
that I think is where I feel more comfortable. I thought what you were gonna say was methylation is the easiest tool to measure in some ways for the things that we do. It may be, even if there's mediation going on, the methylation itself may be a correlate of some other mechanism that is on the regulatory story, right? But it's not exactly that. That worries me because we're not measuring, even when we work with our bench scientists, we may be measuring 3 omics, but we're not measuring the full landscape of
Yes.
Yeah.
That's very important. We should make sure that people in the
Dani, hi. That was a great talk, like all your talks. Just a quick, I'll put it very quickly. One is, I've been surprised that nothing that I've seen has come out yet about you started with the MTHFR and that pathway and its relationship. I haven't seen anything come out of GWAS studies or other places where you might expect it. And if you know, what do you know about that? And then the other question is, I read recently some papers by Robert Waterland where he looks at the Illumina. You may know this. He looks at Illumina assay and he only finds variation among individuals at a certain limited number of places in the Illumina. I wonder if you could comment on that.
Yeah, sure. So for the first one, I mean, I think some of these particular early genotype discoveries and, in a past generation, don't necessarily make it on GWAS hits, which is curious to me. But I think in that particular case of the folic acid supplementation one, if the reverse, if you were just looking at genetic effects on some outcome, you may not see it because it really is this implication that what may be happening biologically requires an interplay between that genotype and some environment or experience, right? And so I think the classic explanation of if it's, that kind of interaction, you might not see it. So maybe that's the answer to that. In terms of the other one, I think we've all, early on, Andy and I were trying to do this too, like, can you, for each spot in the genome, and maybe not CPG, maybe not the right level, it may be region specific, what do you see in terms of within person variability over time, or even over same cell types, versus across person variability? And And I think lots of folks have done both of those mapping, and I think it's true for sure, over time, there's only a subset of the genome or the epigenome that's really changing over time. That has to also be true across people. So that's not that surprising to me that that would be the case.
Thanks.
Nice to see you.
Any other question? I think we have 1 minute left. Wonderful. Thank you, Dani.
Thanks, guys.